Take your seats. Welcome. Thank you very much for joining us this afternoon for a Ken Kennedy Distinguished Lecture. My name is Lydia Kavraki, and I'm the director of the Ken Kennedy Institute. So allow me to say a few words about the Ken Kennedy Institute, and then we'll get started with our speaker. The Ken Kennedy Institute is an institute that has about 140 faculty members at Rice and 170 affiliates. We foster world-class foundational and use-inspired research in AI and computing and enable its transformative use across diverse disciplines to solve real, hard problems, always driven by the highest ethical principles. There are events that we do. We have a lot of things that are going on at the Ken Kennedy Institute. Some are open to the Rice community. For example, our research clusters, our student fellowships, our technical get-togethers. And some of them are open to the community, our conferences, some of our workshops and boot camps, and our distinguished lectures. Just to give you an idea about how the Institute is organized today to support research, I want to bring this map. I don't expect you to read it. I just want to tell you there's a lot of information in our webpage. And what we try is we try to support ground-up efforts for research that is meaningful to our community and to the world. One of our activities is our distinguished lecture series. Today we are going to host Dean Luay Nakhleh as part of this series. Actually, today's talk is the kickoff of our spring semester activities and, of course, of our 2026 series. So I invite you to enjoy the talk, but also to stay connected with us for the two upcoming talks. We are going to have Prabhakar Raghavan from Google joining us in March, and Pietro Perona, who is the Director of Information Science and Technology at Caltech, joining us in April. You can follow us on LinkedIn and our website for updates. So our speaker this afternoon, Luay Nakhleh, oops, I'm sorry about this. I wanted to get back to this talk, to this slide. Our speaker exemplifies excellence in research, teaching, and academic leadership. So taking this in reverse order, Luay Nakhleh is the William and Stephanie Sick Dean of the George R. Brown School of Engineering and Computing at Rice University, and he also holds joint appointments as a professor of computer science and at biosciences. He has served as dean for 5 years. He's renewed for another term, and this is the beginning of the new term. And before that, he served as a chair of the Department of Computer Science from 2017 to 2020. He has been a faculty at Rice since 2004. He continues a legacy of teaching excellence and mentorships for students at Rice, earning numerous awards, including the university's most prestigious teaching award, the George R. Brown Prize for Excellence in Teaching. Today, however, we welcome Luay Nakhleh as a researcher. He is a member of the Ken Kennedy Institute AI in Health Cluster, and his research lies at the intersection of computing and biology, with a focus on developing novel computational methodologies and software to study the evolutionary history of genes and genomes, as well as the genetic relationships among species. So, concluding my introduction, I would like to say that he is the recipient of a Sloan Fellowship, a Guggenheim Fellowship, and he is a fellow of the International Society for Computational Biology, the American Institute for Medical and Biological Engineering, and the American Association for the Advancement of Science. His talk today is going to be on reconstructing evolution across scales, phylogenomics from species to cells. Please join me in welcoming Dr. Luay Nakhleh.
Thank you so much, Lydia, and thank you so much, everyone, for being here, and Happy New Year. I'm glad to see all of you have weathered the blizzard we had a couple of days ago, 40 degrees and sunny, but we cannot handle that, apparently, in Houston. My talk, again, as Lydia said, is about reconstructing evolution across scales. I work on, to me, one of the most fundamental scientific questions about how life evolved on Earth. My talk is going to have the following outline. I will start with This is the Ken Kennedy Institute, is an institute for computing. So I want to actually focus on the computing aspects of the problems, but without getting into the technical details. I try to avoid as much as possible equations and so on, but they will show up every now and then. It's fine. So I want to start by showing you that this is a reverse engineering problem, reconstructing evolution. Then I'm going to talk about going from phylogenetics to phylogenomics in this era, which is when we look at the evolution of species. This is called macro-level evolution, about how all species on Earth evolved. This is what I'm going to talk about, and hopefully you will understand what phylogenetics and phylogenomics are. Then I'm going to go shift scale completely, go down to the individual cell, and talk about tree thinking, this same evolutionary thinking, even though we don't call development evolution. I want to show how we can use the same analogy to think about how cells in our body evolve, and how tumor, how cancer also is a mechanism of evolution. Not a good one, but it is, we capture it thinking about, using evolutionary thinking. And I will conclude with a few remarks that tie all of this together back to computing. So I'm going to start by talking about this as a reverse engineering problem. As I said, for me, one of the things that has always fascinated me is the theory of evolution. To me, the two biggest scientific questions is how did the universe begin? How did life evolve on Earth? And this is one of the most famous quotes by Theodosius Dobzhansky that says nothing in biology makes sense except in the light of evolution. What do I mean by this? If you want to understand how bacteria become resistant to antibiotics, that is evolution. Bacteria evolve and become resistant to antibiotics. Many of you have heard about gene editing. It's a technology that we use today to cure disease, to improve crops. That is the result of evolution. That didn't start as a technology in the lab. That is something evolution created after billions of years of evolution in bacteria. So the CRISPR-Cas9 system is the result of evolution. If you want to understand why certain birds have longer beaks, thicker beaks, and so on, that is evolution. If you want to understand why humans are humans, and the shape of our brain, and why are we the only species that speaks language, that is evolution. So evolution explains everything in biology. And if we want to understand things in biology, we have to shed the light of evolution on them. Now, when we talk about evolution, how we represent it, how we model it, is very, very important. And this is the main character maybe in all of evolutionary biology, Charles Darwin. And this is one of the most famous drawings by Darwin from one of his notebooks over 150 years ago, where he wrote, "I think," and he started drawing this. So many of I assume maybe some of the people in the audience, when we say tree, computer scientists, when we say tree, some of you in the audience don't know what we mean by that, because we don't mean by the trees that you see outside. This is what Darwin was drawing here is a tree. This is the branching process. So what you are seeing there, he was trying to capture how he thought these species will evolve and split and differentiate. And this kind of pattern is what we call tree in computer science. And this is what allowed us also in evolutionary biology is to focus on how we build the tree of life, which is the evolutionary history of all extant species on Earth, all the way back to their most recent common ancestor, which was about 4 billion years ago. Now, we want to reconstruct evolution. It's really reverse engineering the history of life. Why is it reverse engineering the history of life? Because how do we get the data and what it is that we are trying to build? Sequencing machines today, you go to biology labs, they have genome sequencing machines. These sequencing machines get DNA. They get DNA from individuals like us. If they want to actually understand how humans evolved, they will get DNA from us. If they want to understand how primates evolved, they will take DNA from humans, from chimpanzee, from gorilla, and so on. And then we want to try to reconstruct that. The DNA that we are sequencing today is data or DNA that has traveled over millions and billions of years. Okay? And we capture it today, and then we try to reconstruct that. We try to reconstruct the past. We are reconstructing the past from data that we acquired only today. Just think about how daunting this task is. We have data from today. We are trying to reconstruct 4 billion years of evolution. Now, some of you might be aware actually of a different problem that is also reverse engineering, and it has similarities. I like to mention that as an analogy, which is reverse engineering the early universe, right? So there it's not about sequencing machines and the genome, but you have the Hubble telescope. Hubble telescope is capturing lights or photons that traveled for millions and billions of years to get to today, and Hubble telescope captures them. When you capture enough of these photons and enough of this light, now you are assembling an image of the universe as it was billions of years ago. Because this light that is assembled today by a computer program has been traveling for billions of years. Okay, so there is that analogy. One is reconstructing the evolution of life from DNA sequences. One is trying to reconstruct the early creation of the universe, or how the universe was created billions of years ago from light. But there are major differences between them. Hubble captures light that has traveled for billions of years unchanged, and it reached us today, and we are capturing that. So even that photon that Hubble is capturing today, this is from billions of years ago. DNA is not the same. The DNA that we capture in a sequencing machine is from today. It has evolved over time and changed. It's not like light. It has changed over time because of DNA replication, and it carries signals from the past. So if I look at my genome, there might be 1 or 2 letters there or 2 nucleotides that might still be from millions of years ago. Okay? But DNA differs from light because we are capturing it from today with all the changes that have happened. So now we collect all this data, and this actually give rise to this very broad area of phylogenetics, how we build and how do we understand the evolution of taxa. There's that word taxa there, plural, singular is taxon. Taxa can be genomes, genes, species, languages, anything that evolves. We can actually capture its evolution through phylogenetics. But this is typically the inference of these evolutionary histories from heritable data. Okay, the data has to be heritable, right? So that we can actually say this data came from the previous generation came from the generation before it, and so on. So this is what we call phylogenetic tree. Okay, so it's an evolutionary tree. And again, as I said, a tree in computer science is a data structure. It's a very, very fundamental data structure. A little bit of technical here. Just when we talk about a tree, trees have nodes in them. So any of these events where you split something into 2, we call it a node. The tree has a root, a single root. It's unique. And that root has no parents. It has 2 children. Then we have these internal nodes. An internal node has 1 parent, 2 children. Then we have the leaves of the tree, which are the extant taxa or the extant nodes that have no children. So this is a computer science, mathematical data structure that we use to capture the evolution of species. Taxa are these things that we want to infer the evolutionary history of. So in this case, the taxa are a subset of primates. Now, when coupled with the model of sequence evolution, if I add to this tree some mathematical model of how DNA sequences evolved over time, this becomes what we call a generative process. We live in the era of AI, and everyone talks about generative process. Generative process as in I can take this tree now, I can feed it into a computer, I can generate data for you, I can generate sequence data for you. And this is what evolution did. It started about 4 billion years and started generating all this complexity that we see today in life. So what is the phylogenetic tree inference? This inference problem that we want to infer the past. What is this problem and how do we do it? And why am I saying it's a reverse engineering problem? Because if I want to understand the evolution of these primates here, 5 primates, how do we do that? If a biologist wants to infer the evolution of these 5 primates, the first thing they do is they obtain data. The data we collect today is, thanks to advances in biotechnologies, the data we obtain today is genomic data. So you can go get DNA data from human, chimp, gorilla, orangutan, and so on. Okay? So the first thing we do is we collect data. Then we do use computing and mathematics. We need both of them. The mathematics is needed for us to model how we think evolution happened. So this will be the mathematical part. Then once you have the mathematical model, you need to do inference under that, and this is where algorithms and computing comes into the picture. From this, once I have the data, once I have the model, once I have the algorithm, I have the output, which will be the evolutionary history of these organisms. So this is why evolution is the ultimate unobserved process. If you think about generative processes that we deal with, this is the ultimate generative process. It has been ticking for about 4 billion years. It gives us all the data that we have today. It is unobserved. The entire past is unobserved. Sometimes we might have fossil record here and there, but that's basically it. The majority, the great majority of the past is unobserved. We don't have data on it. So this is why we need to reconstruct it. And the reconstruction is a massive problem of missing data. Anyone who deals with statistical inference know that missing data is a big inference problem in statistics. Okay? So this is basically, I wanted to motivate why evolution? Why do we care about evolution? And what is this problem we are dealing with that we want to do this inference? Which is, again, we're trying to take data from today, and we are trying to map hundreds, thousands, millions, billions of years into the past. So the second part I'm going to talk about now is going to get a little bit computational here about phylogenetics and phylogenomics, what it is that we do and why are these computational problems. Okay? Why isn't this a biologist working on it rather than a computer scientist? So in the pre-genomic era, before we had these sequencing machines and we could sequence whole genomes and whole genome sequencing became affordable, what the biologists used to do is that they will get very small DNA sequences. That's what basically was possible and affordable. You will go get four DNA sequences. So here what I'm showing in this example, the data sequence 1, 2, 3, 4, imagine these are 4 sequences we take from 4 individuals. Okay, so the first sequence, ACTGG, this is DNA. There are 4 letters, ACTG. Suppose that sequence 1 that the biologist sequenced from my genome. Sequence 2, they sequenced it from a chimp. They went to the zoo, got a chimp, and they took a sample, sequenced the DNA from that. Sequence 3 is from gorilla, sequence 4 from orangutan, and so on. So this is what the biologists used to do. Now, one thing I'm not going to elaborate on, but I want you to know that the sequences, we want them to be of the same length. Okay? We want them to be of the same length for very important concept underlying mathematics of how we do this inference. We have the data. As I said, the next thing we need the model. What is the model of evolution? So typically, as I said, we have this phylogenetic tree, a tree that, if you look at this, what's happening here, these 4 sequences on the previous slides, they will be at the leaves of the tree. They are today. The past is all these red lines and the green sequences. These are ancestral sequences that we have no access to. We can never observe. So the model will be this tree that I'm trying to infer, to reconstruct. Plus, I need to put some model of sequence evolution. So I need to say, I believe when DNA mutates, it mutates from A to C with this probability, from A to T with this probability, and so on. Okay? So this is where that model of evolution comes into a picture. Mathematically, for those in the audience, the most commonly used tools here is continuous time Markov chains that we use to capture how these sequences evolve over time. So if you give me a tree, this topology, the shape of this tree, plus the branch lengths, how many years have passed between every 2 nodes, plus a model of sequence evolution, that is all I need for me to be able to explain the data. The inference problem becomes like this. Now I have the sequences that someone gave to me. Again, we collected from these individuals, and I want to infer this. I want to infer this past. You know, this could be like 500 million years ago, and this is 200 million years ago, this is 5 million years ago, and so on. In computer science, I mean, this problem is a massive search problem. What most formulations of this problem end up doing, you end up walking in the massive space of these models. So basically you say, is it this model? No. Is it this model? No. Is it this model? No. And so on. This is how we do it. You walk in this space. We say it's actually, we use the word walk computationally. You are walking in this space of the models. This is a massive space, and it has problems for us computationally. It is discrete and continuous. We don't like that in computing, usually. We either like discrete, or we like, of course, we like continuous more. We like discrete second. We don't like discrete and continuous, but this is the problem we deal with here. The discrete part is the tree shape. You know, it's a discrete structure. The continuous part comes from the fact that I have times on these trees. I can tell you the amount of time it passed between two nodes and so on. But the number of trees is massive. Right? So if you give me, let's say, just 80 genomes and say, give me a tree on these 80 genome, the number of trees, if I want to exhaustively explore all the possible trees, the number of trees is a 1 with 139 zeros in front of it. 1 with 139 zeros in it. That's tens of orders of magnitude the number of atoms in the observed universe. Okay? So this is a massive search problem that we need to deal with. And this has been a very active problem for decades now. And the problem is, for the computational people in the room, the problem is NP hard, which means we do not know of any efficient, exact algorithms for the problem. There are hundreds of algorithms because it's NP hard, so we have to do heuristics and approximation algorithms and so on. On the statistical side, every possible tool in the textbook has been tried. Maximum likelihood, maximum parsimony, Bayesian inference, all of that have been tried on this problem. But high performance computing has been critical. If you want to deal with a problem that this massive, with the discrete continuous space, with this size of the space, with this challenge of computing likelihood functions and so on, this is where you need high performance computing. So this is the phylogenetic problem, and this is what we dealt with. So complex, but this is what we dealt with before we had the whole genomes being sequenced. And that's when we had whole genome sequencing, then we moved into the phylogenomic, not the phylogenetic, the phylogenomic era. And what happened there is that a lot of complexity. So when you start sequencing a whole genome, so if someone says, I'm going to sequence your genome for you, think about it like this. Your genome is one word that has 3 billion letters in it. Your genome is one word with 3 billion letters. When they say we sequence your genomes, they are not giving you don't get on a CD, no more CDs now, or USB drive or anything like that. Here is that one word of 3 billion letters. What sequencing machines give us? These short snippets of DNA from your genome. So they give you a big bag of small pieces of your genome. And not only that, they will have errors in them. So there will be a place that your genome has the letter C. The sequencing machine made an error and said it's a T. So now your genome is given to you as these small pieces of a puzzle, a huge big bucket of these small pieces of a puzzle, billions of them. This is the first step when we say we have the genome. Then, okay, I cannot do that. I need the sequence of the genome. I need that word. So we then need to assemble that genome. So that is a purely computational task. That's purely computational task. You need an algorithm to assemble the genome. That algorithm makes mistakes. There's no perfect algorithm for assembly. Okay, so keep in mind this. We started with sequencing. It introduced error. We assembled the genome. It introduced error. And then we need to align the genomes. Remember, I told you that I want the sequences to be of the same length. So if you sequence the human genome, chimp genome, gorilla genome, I want them to be of the same length. So I want you to make them of the same length. That is a very famous problem in computational biology called sequence alignment. That problem is very hard, computationally very hard, and you introduce error. So now, just think about it. I have not even started with building the evolutionary history of anything. I'm looking at data that has at least 3 sources of error in it. Okay? And now I need to build a tree from that and deal with all the complexity. So this is, and I'm not going to focus even on this. You can have talks on all these ones here on sequencing, on assembly. In fact, we have an expert on genome assembly is Professor Todd Triangen, who's sitting in the back here in computer science. And we have lots of other faculty who work on genome alignment, including Todd himself. But we have a lot of expertise in this area at this university. So if we put aside all these issues, error in the data and all of that, what does this phylogenomic problem? The phylogenetic inference was I have DNA sequences, give me a tree. Now the problem is I change one word. I have genomes, give me a tree. I have the set of genomes, give me a tree. So now the question that should be on the mind of any computational person, if I were to give this to a PhD student and say work on it, the first reaction of a PhD student should be, But we have decades of work on phylogenetics where you took DNA sequences, built a tree. If I'm giving you genomes, what's the big deal? Just run it. The sequences are longer. The sequences are longer. So the first question one should ask, okay, is there something to do here for computational people? Or am I just working the same problem, run it? It's the sequences are longer. I understand the method is gonna take more time, but the sequences are longer. I think you know what the answer will be, otherwise my talk is finished. The answer is no, okay? The answer is no because of complexity. And two major sources of complexity I'm gonna talk about. The evolution is not a tree, it is a set of trees within a tree. Okay, so what happens here? And I'll try to explain this. This is really a biological issue here that for us to mathematically model it, we have to understand biology. And this is always my message for people who wanna work in computational biology. You cannot be a pure computational person and say, I want to work on biology, but I don't want to learn biology. You know, it's very important to learn biology because that's how we do accurate modeling. Okay, so this here, what you are seeing is the evolutionary history of human, chimp, gorilla. Okay, so human and chimp, we are closest species to each other, and we split from each other about a few million years ago, and then our ancestor split from the gorilla a few million years before that. Okay, so humans are closer to chimp than chimp is to gorilla, okay? So this is the evolutionary history of human, chimp, and gorilla. Human and chimp are closer to each other, and then gorilla is farther apart. But here's a paper that appeared in Nature, and I'm just highlighting something from the abstract. So when these researchers tried to look at the genomes of human, chimp, gorilla, and they started building trees for different regions in the genome. Remember, the genome is a few billion letters. So they started saying, let me look at the first region, the first gene, build a tree there and see what does it give me human and chimp versus gorilla. Let me go to another gene, build another tree, and so on. What they observed is that that's not true. About 30% of the genome tells you a different story. 70% of the genome told us human is closer to chimp. 15% told us human is closer to gorilla. 15% told us chimp is closer to gorilla. So now you have this phenomenon that you are looking at it, different regions in the genome are telling you a different story, and what's happening there, and how do I then tell what is the evolutionary history, what truly happened. Okay, so what happened here is that this is pictorially depicted here. So what you are seeing in the green is a chromosome or a part of the genome. What you're seeing here, these yellow regions, imagine that these are regions in the genome that we sequence, got DNA from. So when I start building a tree on this region, it tells me human and chimp are close to each other. I build this region here, it tells me human and chimp close to each other. Now it changes. This one tells me human and gorilla closer to each other, and so on. So why does this happen? Why does this story happen? And this is just human, chimp, gorilla. If you take any set of genomes, you are going to see the same mess. Okay? So what this happened, now I need to look at evolution and what happened. When we look at evolution and what happens truly in biology, a human, chimp, gorilla is not one individual in each. Human is a population. We have billions of people. The same thing with chimp, it's millions or whatever the number of individuals. Same thing with gorilla. So if you start looking at it from a population genetic perspective, if you start looking at the populations, what happened there is a very important biological process that happened called recombination, meiotic recombination. Meiotic recombination starts shuffling DNA. It's shuffling DNA, then when you look at the genome, imagine that you are walking along the genome from left to right. And every time you are passing in a region, you are building a tree. You start seeing shifting and switching in trees as you move from left to right. And this is what happens here. So the genome is truly a mosaic of different evolutionary histories. Okay? Now, when we have something like that, okay, how do we infer? So now someone is telling me different regions in the genome have different histories. But I'm interested also in how species evolved. Okay? If someone asks me how human is closer to chimp or to gorilla, I don't say 30% closer to gorilla or something like that. I say, yes, human is a sibling of chimp based on our understanding. And that's where I would say the central formula in all of this comes here, that when someone says, what is the likelihood of a species tree psi, given genomes G, This is now the formula that we changed from phylogenetics to phylogenomics, where we are saying that these genomic regions, different genomic regions evolve down different trees. This is the T. And different T's, these trees, evolve down that species tree. Mathematically, whenever you see this kind of symbol here, the integral, basically what we are saying, we don't care about the gene trees. You can treat them as a nuisance parameter, integrate them out, because I am interested in what was the evolutionary history of species given the genomic data. I'm not looking for the trees themselves. And this has actually led to a massive explosion in research here in the mathematical side and in the computational side, because we need to figure out how do we compute these things, okay? So now the interesting thing for us, if you think about it from a statistical perspective, now you can look at the species tree as a generative process, something that generates data. So if you look at the tree, think about it that I can think of it as a process that will generate gene trees. It will generate trees for different regions in the genome. Every tree, when I couple it with a model of sequence evolution, it also generates sequence data for me. Okay, so this is a generative process that actually can, from a species tree, how the species evolved, I can get down all to the genome. So this is a mathematical model that can allow me to capture how do I observe genomic data from the evolution of species. And this is the formula that can capture the likelihood of it. Where do we get these quantities? I'm not going to elaborate too much on them. This one quantity, the only thing I want to say, PLGT, one of the most classical results in this field is a 1981 paper by Joe Felsenstein from University of Washington that showed how you compute this polynomially. For those in the audience, computing the likelihood of a given phylogenetic tree actually is a polynomial problem, and this was a very, very innovative algorithm. In the phylogenomic era, this is the one that actually gave us hard time. What does it mean to talk about the probability of seeing a gene tree given that species tree? And this is where we needed to start looking mathematically, start looking at these trees evolving, thin trees evolving inside thicker trees. Or in other words, if you look at this, and this is an evolutionary tree that says human and chimp are closer to each other than either of them to gorilla. This is not how we should be looking at this tree. If we want to understand why gene trees are different, we need to look at it like this. Okay, so we need to look at this, that humans is a population, and chimp is a population, and gorilla is a population, and every dot here is an individual in this population. Every row of dots is that population in a given generation. In today's generation, these dots are us. The previous generation, think about it 20, 25 years, 30 years ago, this was the previous generation, and so on. Once you start looking at it, we can start now quantifying mathematically what are these probabilities, what is the probability of me seeing these kind of gene trees. And I'm not going to read them here, but this is for a human chimp gorilla. I can actually tell you exactly the probability that you will see human versus closer to chimp or human closer to gorilla, and so on. The problem is that biology is much, much more complex than this. That's one process that leads it. Another big process that leads to this kind of phenomena is what we call gene duplication and loss. So you start having these genes duplicating in the genome and giving these kind of patterns. So this becomes more involved. Again, I'm not going to go through the mathematics of it, but this is some very, very open research areas. So the problem is very hard. Likelihood methods, like computing the likelihood, is very challenging, but we have to make progress on it. The biggest open question here, in my opinion, is how do you combine multiple sources of what's happening? How do you combine this gene tree incongruence with gene duplication and loss and so on? Because in biology, that's what's happening. For us, mathematically and computationally, we like to separate them because we want to make our life easier. But that's not what truly happened in biology. In biology, all of it, all the complexity happens at the same time. But the second level of complexity is not even trees within a tree, it's trees within a network. Okay, so biology keeps getting more and more complex. Okay? What do I mean by within a network? If you heard about bacteria, bacteria don't evolve nicely. Okay, they don't evolve down a tree cleanly and so on. They exchange a lot of DNA. Okay, bacteria exchange a lot of DNA across species boundaries. In fact, this is one of the mechanisms by which bacteria can become resistant to antibiotics. So imagine you have bacterium, you have another bacterium that happens to be antibiotic resistant. they can actually swap DNA. Okay, different species, they can swap DNA. There are lots of evolutionary processes that are similar to that kind of horizontal gene transfer in bacteria. Hybridization is very big in plants. So over 25, 30% of all plants are hybrids. Hybridization means mating of individuals from different species. So you can no longer use a tree to model them. You have hybridization in mosquitoes, you have hybridization in butterflies, you have hybridization between human and Neanderthals. This is a very famous story about the gene flow from Neanderthals to humans, in particular to the European subpopulation. You have hybridization in primates. So if you look at this, this is from this paper in Science that appeared a couple of years ago. This is the evolutionary history that was presented by the authors in that article. If you look at it, it's not a tree, okay, because it has this kind of node here that has two parents, because hybridization happened in these species, where DNA is from different species, okay? So when this happens, the evolutionary history is no longer a tree, it's what we call a network. A network is something like this. Okay, so the main difference between a tree and a network is this, that we have these reticulation nodes, that you have nodes that have two parents. So now the DNA that is inherited from two different parents. And this is where we start having phylogenetic networks. And similar to before, again, when I look at it statistically or mathematically, this is a massive generative process. The phylogenetic network, this network generates gene trees for me for different genomic regions. Every tree with the model of evolution gives me different sequences. So now I can tie genomic sequences all the way to this phylogenetic network. Again, in the machine learning language, this process is actually a mixture of generative models as well. The formula, again, this is the beauty of math, it did not change. It did not change the likelihood thing. The only difference is that this psi now is a network, it's not a tree. This is really the main difference between them. There are lots of computational questions that are open here. How do we incorporate multiple sources of incongruence? How do I think about hybridization and gene duplication and loss and so on? Because again, that's what truly happens in biology. When we computational people decide to ignore duplication and loss, let's say, we are doing that just for a simple reason. We want to make our life easy, and we want to be able to develop algorithms. Biology is not simple like that. I cannot rule out any processes. Taming the complexity of likelihood calculation. So here we go into an area where the likelihood calculation is intractable. We don't have polynomial time algorithms. This is something that big. Large-scale inference is beyond the reach of current methods now. You know, we have actually, my group, we have been developing tools for several years now. We can infer these evolutionary histories, but for tens of genomes. Biologists today have hundreds and thousands. We cannot analyze that at this point, given the complexity of these problems. A massive problem here for us is to network or not to network. That's the question. The model selection. Networks are very complex models, actually arbitrarily complex. And when you have arbitrarily complex models, that's a problem, because a model with 2 parameters is better than a model than 1, fits the data better. A model with 15 fits better than 2. There's a famous quote that with 5 or something like that, 5 parameters, I can fit an elephant. With 6 parameters, I can make it wiggle its trunk. This is the problem that we deal with here. Networks are very complex. So when you start inferring them, it's very important to say, are we inferring biologically realistic things, or are we just making the model more complex? Because the more complex, the better from a mathematical side or from a fitting side. So this is a very open question for us. So that was about the macro evolution, species and stuff. Now I'm going to shift scales completely, not millions of years, not billions of years. We're going to go now to the cells in our body. And I want to look at it, at the development and stuff. And it's going to be shorter part now. I'm going to talk about how we use this tree notion to think about cells in our body and cancer. We don't talk about cells in our body as an evolution, right? Nobody said we talk about development. We don't talk about cancer as evolution as well. But this is the beauty of math and computing, that you can abstract all of them to use the same tools. So what happens with cells in your body here, okay? So when you have, what happens with cells is that they divide and differentiate. Your cells are always dividing and differentiating in your body, okay? So your cells, in fact, are supposed to die, and you are supposed to get new cells all the time, and they are dividing all the time. Cells are dividing all the time, and they differentiate, so you start having different types of cells, okay? But once you have that, division and differentiation, that immediately the word that comes to mind is tree. Okay, that's a branching thing, so that we can capture with a tree. So now the tree is not, human, chimp, gorilla, whatever. These are cells from our bodies. These could be cells from a sample of cancer tissue. These could be cells from a neuronal segment in our brain, or anything. And if we try to understand how they split from each other and try to infer the past of these cells, we can use trees to capture that. So one of the major problems in this cell biology here is what's known as cell lineage tracing, where you have these cells. Once you build the tree, you want to actually try to trace back the types of the cells that gave rise to these ones. And that's typically done from either mutation data or some today from CRISPR data, okay? Some barcoding data that we can do. So this is a computational problem that operates on trees. The trees are of cells, not of species. The other problem is trajectory inference, is that how cells change from one type to another, right? So cells change types over time. And these are some of the problems that we do. I'm not gonna go into any details here or talk about similar level that I talked about before, but these are very computational tasks. There are major computational challenges here. Scalability to millions of cells. Today you have data, biologists can get access to data on millions of cells from an individual. How do you do this type of analysis at that scale? Uncertainty quantification. The data that you're observing from here has a lot of error. So how do we sift through that? Am I looking at error, or am I gonna look at mutation? Because I need that signal for me to be able to build that. Data integration is very important. One source of data, let's say the mutation, can give me the tree. Another type of data, like transcriptomics, RNA, can tell me something about the trajectory inference. What if they don't match? How do we reconcile them? So we need to integrate the data and do some simultaneous inference. So these are some of the computational challenges in this area. But one of the beautiful things I've seen, actually this paper by a famous evolutionary biologist, Jeff Doyle, where he actually talks about this analogy between the concept of species and cell type. So what happens in species? How do we have species over evolution? I mean, there are lots of issues with that and wrong depictions of them, which is why some people have some issue with it. But the way speciation happened, imagine that you had one species and for some reason, migration, ecological event, and so on, the population split. And these two populations now start being isolated from each other, diverging, evolving independently. At some point, they become reproductively isolated, and that's where biologists like to call them species. But there is that gray zone in the beginning when these two populations split. They are still very similar. So there is that gray zone where we look at them and say, are they two different species or are they the same species? At some point, it becomes clear they are two different species. The same thing happens with cell types. When a cell divides and differentiates, and we start looking at two cells, there is that gray zone in the beginning where we say, are we looking at two different cell types? At some point, yes, become so clear, right? So clear the cells at what they are, but there's that gray zone where we don't. So we can draw these analogies, even though evolutionary biology looks at species, not at cells like this. We can use all these tools, mathematical tools, computational tools, to understand how cells evolve or divide and differentiate and how cell types arise. Now, one word about species and cell type. I'm not going to get into a long discussion about this. So species is actually maybe the central concept in systematics and evolutionary biology. And cell type is a major goal of cell biology, understanding cell types. The interesting thing about this is that biologists till today do not agree on what is a species and what is cell type. Okay, I just wanted to say that. Then it becomes, even though they don't agree on that, there are some computational formulations for them. So we have species delimitation problem. That's actually a computational problem where you feed the algorithm a set of DNA sequences, and the algorithm comes back and say, these 2 sequences are the same species, these 5 are the same species and stuff like that. This is known as the species delimitation problem. You have similar one to cell type as well. One of the issues there that we take these two concepts that are not defined by biologists, and biologists will argue about them forever. What is a species, what is cell type? But then the computational technique that actually permeates most of the techniques for solving these problems is clustering based. You know clustering, how you take sets of points and cluster them. But here is actually the funny, interesting thing about it. We have 2 biological concepts that are not well defined. We try to solve them with a mathematical or computational task that is not well defined either. Right? So clustering is either not that well defined, actually. So the notion of cluster cannot be precisely defined. Clustering is in the eye of the beholder. Okay? So it's a very interesting thing. You know, we have, and this is the messiness of biology and the beauty of it, honestly. Okay? Biology is not about mathematical theorems and proofs. There is no theorem and proof in biology. Okay? But it is messy, and to me, this is why it's beautiful, and I like to work on it. I'll finish just with a little bit about in these individual cells and development, what happens when the tree of these cells goes crazy, and that's cancer. So what happens in cancer? Today, when a cancer patient shows up, they take a biopsy from that cancer patient. So you take a biopsy and you can start sequencing the genomes of cells in the biopsy from that patient. And one of the things that have been observed today, that when cancer patients show up and you take biopsy and you start taking these cells and sequencing their DNA, the biologists, oncologists are noticing you are starting to see different groups of cells with different signatures, mutations in them. Okay? These groups of cells here have mutation in position 1 in the genome. This group of cells have mutation in position 5,000. This group of cells have a different mutation. So they are observing this, which is known as intratumor heterogeneity, that when you look at the tumor from the same patient, you are seeing different cells have different mutational signatures. Now, we can look at evolution as cancer as an evolutionary process, because cancer is a somatic evolutionary process in a population of cells. So what happens here? Here I'm showing time from left to right. So here, if I think about these yellow dots as cells, healthy cells in an individual, one of the things that happen, everything is fine, healthy cells, at some point, the wrong mutation hits. Okay, the wrong mutation hits. What happens here is that what I'm depicting by this color now dominating here is that these cancer cells start taking over the population. Okay? In that region, you start having this mutation spreading. Because remember, cells are going to divide and differentiate. So that mutation that was there once, now when you have 2 copies of a cell, you have 2 of these, 2 cells with that mutation, then 4, then 8, then 16. This is what happens. Then at some point later, another mutation hit, and so on. So when you take a segment today or a biopsy from the patient and you sequence it, you see these different colors are depicting different groups of cells in the patient with different mutation. And this is how we explain intratumor heterogeneity from an evolutionary perspective. Now, I'm not going to elaborate much on this because I'm almost out of time here, but there are different modes of evolution that we can capture and assess from the shape of the tree. Okay, so when we construct a tree, there are different shapes. This shape is different from this. This shape is different from this. These different shapes have implications on diagnosis, prognosis, and therapeutic treatment as well. Depending on the shape, what this tree is telling us about the cancer, it sometimes tells us you need 1 biopsy is sufficient. Sometimes what's happening today is something called spatial transcriptomics, where you take data from different regions, depending on how things have been evolving. Sometimes all you need to do is target a major mutation. Sometimes you have to target many mutations. So this is where the use of these trees can help with these areas of diagnosis, prognosis, and so on. So how do we build these trees to answer these questions? Today, one of the great developments in sequencing technology that we can sequence individual cells. This is called single-cell DNA sequencing. Okay, so 15 years ago, when you heard about sequencing, or 25 years ago, when you heard about sequencing the human genome, we didn't sequence individual cell in a human. It was an average, right? So you take a whole set of cells, You sequence them and you get the average DNA from an individual. There is nothing wrong with healthy cells. With cancer, it's problematic because that average is missing the point. So in DNA single-cell sequencing, you can get DNA from individual cells. You sequence them, you assemble them, similar to before. And then from that, our task becomes, once I have the genomes assembled, I want to find what mutations happened in what cells and what's the tree structure that explains what happened in this patient. The issue here is that, again, this is, I said before, that you need to understand biology and respect it if you want to develop computational tools for it. But also you need to understand the biotechnologies and respect them as well. The data we collect is not accurate, is not correct because it's coming from a technology. Technology in single-cell sequencing makes several mistakes. This is where that conversation happened between the modeler and those who actually develop these technologies or use them to understand what are the types of errors that happen. And this is what we did in this project. In our collaboration with MD Anderson, our colleagues told us these are the types of errors that the sequencing machine can make. Then we said, okay, now we understand about the error. We understand how cancer evolves and stuff like that. Now our inference problem, we want to infer a tree. This tree is different now. We need to infer these clones or clusters of cells with different mutations. I need to infer what mutations actually happened. And I need to do all of that while accounting for error in the data that the biologist told me the sequencing machine is making. This is one of our methods that we developed here. Again, for me, I'm not going to read this, but the point is, once you have a clear understanding of what's happening, the modeling, it looks complicated. All this is saying in English. This is the data that I got from a sequencing machine. It has two types of errors. The data that I'm seeing came from some DNA data that I never observed. That DNA data that I never observed came from a tree and a model of sequence evolution that I never observed either. And then there was some sort of this clonal structure that there are different clusters, right? So if you know how to read these diagrams, these are called graphical models, the rationale for it is very straightforward once you understand how cancer evolves, how sequencing technologies work. Now, once we had this, we did inference under this model. We analyzed several cancer data sets. For example, this analysis of metastatic colon cancer data set. So we analyzed it. We got the tree structure from it. And we ran another tool on it. And one of the things that we discovered, for example, that in this cancer patient, the cancer actually had metastasized to the liver from the colon through different pathways. Okay, so this was very, very nice information that without this computational reconstruction, we would not have gotten. The data you observe is from one data point that you cannot get. There are lots of open computational questions here. I'm not going to elaborate on them too much. I'm happy to talk to anyone, especially on the computational side, who are looking for collaboration in these areas. We are always happy to chat about that. But I just want to end with a few concluding remarks that, to me, when I think about it, as a computer scientist, I've been in the faculty for 21 years, when I think about what is the most powerful tool that computational people give the other sciences, I would say it's the abstraction. I think abstraction is the best thing that we contribute to the field. What do I mean by abstraction? Biology is very complex, So if you look at it, if you are studying the history of life, evolution of all species on Earth, if you're trying to understand HGT, horizontal gene transfer of antibiotic resistance, cellular differentiation, clonal expansion of tumor, not only are these different biological processes, these are different biologists who study them. There's no biologist who sits up their lab and one day they are studying evolution of life, next day bacteria, third day cancer. Biologists are much more specialized than that, right? So biology separates these things. There are tumor, there's antibiotic resistance in bacteria and so on. But if you think about it mathematically, one thing unifies all of them, the tree structure. Okay, so tree structure, which is a mathematical structure, is actually fundamental unit. I can actually capture how cells divide and differentiate, how cancer evolves, how species evolved on Earth, how bacteria became resistant using trees. Even networks, I didn't want to go into the details. A network is just a mixture of trees. Okay, so I can capture still networks with a mixture of trees. So this is, to me, one of the most beautiful things computationally, mathematically, about thinking about these problems. How do I bring seemingly disparate problems from biology and using the power of math and computing to unify them? Scale invariance, the mathematics of these models that I showed, like the multi-species coalescent or branching processes, which are heavily investigated in statistics, is scale agnostic. Whether I am running it on millions of years because I'm studying species, or whether I'm using it for days or weeks because I'm looking at cells in an individual, they are the same. It's the same math. The equations do not change. The computational frontier, one big thing here, while the math doesn't change and so on, signal to noise is very important. Distinguishing biological processes from technical data artifacts is one of the most ubiquitous problems in biological data analysis. All data you get is the output of a machine. These machines make mistakes. When you look at these mistakes, remember that mutation is also a mistake. So are the mistake you are looking at, is that from the machine? Or is that a mutation that happened during evolution? That's a big thing for us. Signal to noise, model identifiability. This is also a very big issue in statistics. When you're trying to infer a model, say, this is how life evolved, this is how this tumor evolved, how sure are you of that? Right? Because what if another model fit the data equally well? So this is known as model identifiability. It's a very central issue we think about. The last thing I want to say is that this is a big data problem, coming from the mother of all generative processes. Evolution on Earth actually has been a generative process, as I said, working for 4 billion years, but the ground truth is completely missing. We don't have the ground truth. So this is why this is the generative process where we have data from today. We are trying to reconstruct that entire history. This problem is an inverse problem of massive scale and massive complexity, which requires high-performance computing and high-performance algorithms. And this is why I find it super fitting, given that it requires high-performance algorithm, that I was invited to give this talk at the Ken Kennedy Institute, which is named after Ken Kennedy, a former professor in our computer science department here, and who's one of the pioneers of high-performance computing. And with that, thank you very much.
Thank you. Thank you very much. We have time for a few questions, and we are going to pass the microphone when you have a question so that our audience can also hear you. All right. Okay, I think we have one question over there. Microphone is coming to you.
Thank you. Thank you for your talk, and I like your little slide setups. So I don't know much about cancer, but in your experience, there is like an incongruency between taxonomy and when it comes to species and phylogenetics, right? Sometimes certain species are not, they might have the same genus species, but they're not necessarily closely related. Do you see cancer being better at that, with like different stages being classified perhaps more specifically, that they do reflect, that is reflected in the phylogenies that are obtained?
So if I understood the question correctly about species, is sometimes there is lack of clarity. Are we looking at different species? Again, as I said, biologically, the concept of species even is, nobody is confusing human versus a tree outside, right? But, it can get complicated in other domains. When it comes to cancer, we don't have that similar analogy of, like in the cancer specifically, we are not looking at the types of them. Where the analogy comes in the area of cancer, when I talked about these clones or clusters, again, keep in mind a few slides before that, I said clustering is not a well-defined problem. So when we get this, so I've seen papers, let's say, I'll show you, can Google it and find it, very top papers in Nature and these kind of journals. They talk about concepts like clone, superclone, subclone, stuff like that. I still cannot find a mathematical definition of what these are. So there are these issues there. Biology is a fuzzy field, right? Again, it's very, very hard. And this is, to me, one of the challenges, that even with problems like species delimitation and cell type, if you cannot define it mathematically, I do not believe you can infer it computationally, right? So there are these challenges there. So we have to put some sort of definition, even if they are fuzzy, and work with them. But I would say that problem of determining are we looking at different species or not is not there in the domain of cancer.
Thank you.
Okay, we'll take one more question up there. Which one? Lady up there.
Thank you so much. I love what you said at the end about abstracting the biology. Can you talk a little bit about how you identify the different biological, I guess, areas of research that might fit your methodologies?
Oh, I don't try to find problems that fit my methodology. Right? Because I find that not an interesting computational approach for doing research. Again, that's the approach of having the hammer looking for nails. I follow my passion. Evolution is my passion. Even though as a computer scientist, anything that evolves, I'm interested in it. I'll give you an example that I didn't talk about. Actually, I did some research for multiple years and published papers on the evolution of natural languages. So I studied how Indo-European languages evolved and how they, because there is, if you think about this concept of horizontal gene transfer in bacteria, there is that thing between in languages, right? We borrow words from each other and stuff. But when you start looking at languages, how they evolved over thousands of years, how they evolved, when they borrowed and stuff like that. And today, for example, to me, one of the most intriguing questions, can we look at the evolution of languages? Can we look at the evolution of individuals, humans? Because we have the DNA on them, and can understand what happened there. Because language should track populations, right? I mean, if nobody in the United States speak Chinese, you will not see it here, right? But if someone speaks it, you look at the DNA and so on. So it will be interesting to look at that. But this is my approach to research, is that I don't have a mathematical solution or computational solution, and I'm looking for problems. I am looking at problems that intrigue me, and I'm saying whatever the solution. So if you look, actually, this was not a laundry list of our work, but some of the techniques we have are graph theory. Some of them are Bayesian inference. Some of them actually are machine learning. Like we used actual neural networks in some areas there. So for me, the solution, the technique is not my main priority. My main priority is that what's the biological question that interests me? And all of them have that unifying theme of evolution to them.
Okay, on this note, let's thank our speaker. We are going to close the session here.